So in this next section, we're going to be taking a look at some subcategories of our triglycerides. And we're going to lump them together as these molecules known as phospholipids, right? So phospholipids sound similar, but they also sound slightly different. And the key thing about your phospholipids is that they belong to the family of lipids, and they're similar in structure to the triacylglycerols or the triglycerides, but the key difference is this part of the name here, right? So this suggests then that they're going to contain phosphorus, but not just phosphorus, they're actually going to contain phosphate as part of the structure. And what that does, as we'll see down the road, is it actually gives these phospholipids a very, very important characteristic that allows it to carry out their functions, whatever that function happens to be. We'll take a look at some subcategories of these phospholipids. So the names that we're going to talk about here, they may sound really, really big and confusing, but if you break it down into the different parts, they will make a little bit of sense, or maybe a lot of sense. So the first one here we're going to take a look at are known as glycerophospholipids, right? And again, if you look at the name, right, they belong to the family of lipids. Okay, now we know what the phospho part of it means. All right, contains phosphate. And then glycero, right, would mean that there's glycerol present in the molecule. All right, so if you were to draw a sort of a block diagram of what a typical glycerophospholipid would look like, there is your glycerol backbone, which we've seen a lot of already, right? So that forms the backbone of the structure. And then, as we also have seen before, right, we can attach our fatty acids to that glycerol backbone. And what is the glue that sticks the fatty acid to the glycerol? Well, that's your ester bond. And typically in a triglyceride, we would have a third fatty acid down here, but now instead of that third fatty acid, we have that phosphate group. And that is attached to the glycerol backbone by a different type of an ester called a phosphate ester. And there's also a phosphate ester bond on the other side of it right here that attaches it to another part of the molecule known as an amino alcohol. All right, and then this name also tells you what it's composed of. There's an amine group and an alcohol group present in that part of the molecule. All right, so your glycerophospholipid, they look really similar to the triglycerides. The big difference is in this, in this third attachment here. It's not a fatty acid. Instead, it is a phosphate attached to an amino alcohol. So another type of phospholipid that we're going to see is something known as sphingomyelin. Okay, and sphingomyelin, again, it's maybe a tongue twister. The main difference between this and your typical glycerophospholipids now is instead of glycerol, the backbone is now a molecule known as sphingosine. All right, so sphingosine forms the backbone. Right, that's why we have this first part of the name here in sphingomyelin. So it forms the backbone instead of glycerol. It is still attached to one fatty acid by an ester bond. All right, so we're going to have that ester bond here. And the other part of it is going to look similar to what we just talked about previously. We'll have a phosphate attached to the sphingosine backbone by a phosphoester bond, or a phosphate ester bond. And on the other side of it, we have an amino alcohol. All right, so again, there's some similarities, but there are also a couple differences between the glycerophospholipids. So we've talked about this amino alcohol group a little bit, but not in a lot of detail. So let's take a closer look at that amino alcohol. These, as we've seen, are found in not just in glycerophospholipids, but sphingolipids as well. The most common ones that we'll see are choline, which you may have heard about. There's serine, an S is missing here, serine, and there's also ethanolamine, right? Those are the three common amino alcohols that we're going to find in these types of molecules. These are ionized at normal pH of the body, which is about 7.4, right, if you remember back from chapter 10. And what this means, right, this is key, this is extremely important, because what this means here, the fact that they're ionized, it means that this part of the molecule is charged, right? Ions, if you remember, are things that have charges associated with them. So if you look at your structures here for choline, serine, and ethanolamine, right, you'll see that they all have that positive charge in common. Your serine, for instance, also has a negative charge in there. So the fact that these, where these are found in the molecule, are going to be charged has a huge impact on some of the characteristics of these molecules, which we'll get to in a second. All right, so we've talked about our glycerophospholipids that have that glycerol backbone, and they also have a couple fatty acid tails attached to them. And then the third thing attached to that glycerol backbone is going to be your phosphate. And then we have some sort of an amino alcohol, which is highlighted here in green. For this molecule here on the left side, this is a molecule known as lecithin. And the amino alcohol here is choline. And the structure on the right, I'll just draw a line all down the middle. The structure on the right here, again, has that phosphate group that connects the glycerol backbone to another or a different amino alcohol called ethanolamine. Right, so what you'll see here, and I'll try to highlight this in yellow, is that the fact that these amino alcohols, as well as the phosphate group, are ionized, means that this part of the molecule highlighted in yellow is going to be extremely polar. All right, so we have the yellow highlighted part of the molecule here. So the polar part of the molecule, and then the rest of the molecule is nonpolar. So you have a special case happening now where you have these glycerophospholipids that have both a polar, highly polar region, and the remaining part of it is going to be non-polar. All right, and we'll take a closer look at what that means in a second. So lecithin and cephalin, like we saw here, two types of these glycerophospholipids. You find them, cephalin, for instance, in things like brain and nerve tissues. Lecithin is found a lot in egg yolk and wheat germ and yeast, for instance. So we looked at the structure for these glycerophospholipids, and the key thing now is the polarity, right? So the glycerophospholipids are going to have both a polar region, right, which is the region here highlighted in yellow that I'm circling now in red, as well as a non-polar region, right, the rest of the molecule highlighted in green, that will allow this to interact now with both polar and non-polar substances, right? And that's an extremely important characteristic of your glycerophospholipids, right? They can interact with both polar and non-polar substances. Because they do possess both a polar region and a non-polar region. So if we were to, draw like a simplified, well, maybe not so simplified for right now, we'll just take a look at the structures, then we'll simplify it. So we have here a typical glycerophospholipid shown on top. All right, so let's well, that's the components. Let's focus in down here on an actual glycerophospholipid. So let's identify the different regions. There's the glycerol backbone. There's 3-carbon backbone. Okay, attached on the right side here, these are the fatty acids. Fatty acid tails, which remember are long carbon tails, and that means that these are going to be highly non-polar. And then on the other side, we have your phosphate. And then you also have your amino alcohol. Which is choline in this case. And this region then in blue is going to be highly polar. Right, so what you have is a molecule that has sort of a polar head, and there are 2 non-polar tails attached to it. And the most common notation you'll see in your textbook and online is you'll see it being represented like this. So for a typical glycerophospholipid, or just phospholipid in the general sense, we'll draw it like this. You have a polar head, and then you have 2 non-polar tails attached to it. Okay, and this is a really simplified way to look at these glycerophospholipids, but it does explain a lot in terms of how these molecules are going to behave when they are in contact with each other, but also when you put them in water, for example. So the other type of glycerophospholipid we saw was sphingomyelin, and here we have this molecule called sphingosine, which forms the backbone instead of glycerol. And then what that actually does is it forms an amide bond to a fatty acid, not an ester bond. And then the hydroxyl group from sphingomyelin forms an ester bond with a phosphate, which then forms another ester bond to an amino alcohol like choline or ethanolamine. Sphingomyelins are very common. The last name here, myelin, tells you that they actually make up part of the nerve cell, specifically the sheath or the covering around the nerve cell that actually leads to the nerve impulses being propagated at a very high speed, right? So those myelin sheaths, which are made up of sphingomyelin molecules, are extremely important. It allows you to respond to different stimuli very quickly, as if compared to if you didn't have those myelin sheath present. All right, so here's the typical sphingomyelin molecule. So let's identify the different regions of it. So we'll highlight in yellow the backbone. So this is the backbone of the sphingomyelin, which is the sphingosine. So the yellow here is the sphingosine backbone. In blue, we're going to highlight the fatty acid. So here's palmitic acid or fatty acid. And it's connected to the sphingosine backbone now not by an ester, but actually by an amide bond, which is a little unusual, right? So we'll do that one in green. Okay, so we have our amide bond. Right, typically in the previous examples, we saw the fatty acids connected by ester bonds. Here in sphingomyelin, it's attached by an amide bond. And then underneath, we have the phosphate group. We'll do that one in, let's do purple. So that's our phosphate. And that phosphate is attached to the sphingosine backbone by a phosphoester bond. So we call it a phosphate ester, and it's also attached to the amino alcohol on the other side by a phosphate ester bond as well. All right, so this part here is your amino alcohol. Right, again, what you will see is that the region here that I'm going to circle in black, right, is charged. That it's highly polar head, and then the remaining part of the molecule then is going to be non-polar. All right, so if we put a box around the remaining part of it, we have a non-polar region. Right, so we have a similar type of structure compared to the Glycerol Phospholipid we saw previously. So we'll end this subsection by looking at what actually happens when you have these Glycerophospholipids and they are in a polar solvent like water. Right, remember that water is polar. Right, and we're going to draw our Glycerophospholipids. If you remember, there's a head and then we have our tails. And recall here that the head is polar and the tails are non-polar. Right, and the key thing here that's going to drive what's going to occur is this very simple rule we talked about a long time ago, which is like dissolves like. All right, which just simply means that polar things want to be next to other polar things and non-polar things want to be next to other non-polar things. So when you have a sample of these glycerophospholipids in water, for example, the glycerophospholipids will self-assemble into spherical structures called micelles. All right, and the way that the micelles are going to look is going to be driven by this whole like dissolves like idea. So if I have my several of these glycerophospholipids molecules, what I'm going to see, if I were to draw a cross section around them, they're going to look like this, and then their tails are going to extend into the center of this structure. All right, and that is what a micelle actually looks like. Right, and the way that it's arranged should now make a little bit of sense to you. So imagine that this thing is in a big bucket or container of water. Right, so we have a bunch of H2O, which remember is polar. Right, so what you'll see with the micelles which are made, we're going to self-assembly of these glycerophospholipids, is that the polar heads are on the outside, right, where they contact the polar H2O, and the non-polar tails, which we'll highlight here in yellow, right, that forms the middle or the center of the micelle. So the non-polar tails, they don't want to be close to water, so they want to be away from water as much as possible. So the non-polar tails are away from water, and they're actually next to each other, right? Because they have similar polarities. Right, and that sort of goes along with this whole like dissolves like idea. So they're shielded from the polar H2O. Right, and that's the basic idea of how these molecules are going to self-assemble in an aqueous solvent like H2O, right? They're going to form these micelles. The outside of the micelles are going to be made up of the polar heads of the glycerophospholipids. The inside of the micelle is going to be the non-polar tails that want to be next to each other and away from H2O. We're going to come back to this idea of the assembly of these phospholipids when we talk about cell membranes at the very end of the chapter.